yeah, as Antonio mentioned, we're working for HÜBNER Photonics. So VALO Innovation is a part of HÜBNER Photonics. And yeah, we're concentrating on almost all kinds of lasers in the group. So we can talk about single frequency lasers, narrowband lasers. We're doing tunable lasers, beam combiners, and we are also doing femtosecond lasers in the group. And today I want to talk about lasers, or especially femtosecond lasers for simultaneous 2 and 3 photon microscopy. And I will just show you how it works and what are the benefits. So as a brief introduction, so we're talking about microscopy, we're talking about application in tissues. So the optical microscopy, as you all know, can achieve a very high resolution, a very high contrast, and you can get minimal photo damage and maximum penetration depth. The application examples, or typically ones, are the visualization of tumor structures. I will show you some applications later. You can see cell-to-cell interactions, and you can also do in vivo detection of signaling pathways. And I will just explain you in a few seconds where the benefits of using very short pulses. So traditional or linear fluorescence microscopy is in general limited in the penetration depth and also causes photo bleaching and phototoxicity. And you can overcome these mechanisms by using nonlinear excitation techniques. And I will just show you what we are talking about. So we're talking in general about multiphoton microscopy, which means we want to make use of multiple photons at the same time. So we want to have a look on 2 and 3 photon fluorescence processes. We want to have a look on third harmonic generation, and we also can do second harmonic generation. In general, you can see different things with these different methods. So for 2 and 3 photon fluorescence, you can see cell cytoplasm and elastin. Third harmonic generation is always generated at the change of refractive index, so you are able to see cell morphology, you can see the cell structures. And for the second harmonic generation, you can use it to visualize collagen fibers. As we're talking about short lasers, or as you will see, we're talking about pulse duration in the range of 30 femtoseconds. You can just see that, or you can just imagine that the shorter the pulse, the higher is the probability that you have 2 or 3 more photons at the same time. This is necessary to do multiphoton application because, let's say, only if the photons are exactly at the same time at the same place, these multiphoton absorption can occur. So you see that the probability to having 3 photons is for sure smaller than for 2 photons, and you just need to imagine that the pulse duration is one of the important roles here. You can see here on the left side 2 different pulses with different durations. And I just want to say that the area is more or less the same. So you can see that if you just squeeze the photons in time, let's say, to a very short pulse, you just increase the peak power. And so you get a very, very high peak power pulse. And if you just think about comparing a 200 femtosecond pulse to a 40 femtosecond pulse for 2 photon absorption, the efficiency is roughly 5 times higher if you decrease the pulse duration by 5 times. As for 3 photon processes, the process scales to the square. You can just get a 25 times higher efficiency if you just reduce the pulse duration. If we're talking about having the same 2 photon fluorescence or the 2 photon efficiency, then you are able to also reduce the thermal heating by a factor of 5, because you can just simply decrease the average power by a factor of 5, only by having a shorter pulse duration. And then there are different other, let's say, mechanisms where you can reduce the photo-induced damage, and you get a higher contrast and resolution. Just to illustrate what happens, you can see here on the left side, this is a calibration grid, which was illuminated with a 50 femtosecond pulse and around 5 milliwatts, and you see a very high contrast. If you're just using the same power, or at least the same power, but 160 femtosecond pulse, you see more or less nothing. And this is the main advantage of using really short pulse duration, to increase the contrast in the end. On the right side, you can see, let's say this is a scaled up version of the figure in the middle. but you see that the contrast is even smaller. So if we talk about 2 and 3 photon or multiphoton processes, the question is, what are the laser requirements? So from our side, I would say you want to have a femtosecond laser with a very low pulse duration. You need to have very clean pulse because you want to avoid the background, which simply heats up the probes. And this also means that you want to have a very high peak power pulse. As I mentioned before, you are able to reduce also average power to get by keeping the same contrast. And this also allows you for long-term imaging of living cells. And this is one of the advantages we have. Finally, you want to have a very good beam profile because you can get a very small focus in the end. And as you know, I think you need to have some kind of dispersion compensation because simply as we're talking about very short pulses, we're talking about a very broadband spectrum just related by Fourier transformation. So the shorter the pulse, the broader the spectrum. So the more dispersion effects play a significant role here. So you need to be able to control the pulse duration at the sample. Yeah, and finally, for integration, you want to have compact systems. There should be no water cooling, so a completely passively cooled system would be fine. And I will just show you that it will work with the laser we have developed. And this is one of the examples what we are able to provide at the moment. We are able to generate around 30 femtosecond pulses with a very, very low background, which, as I mentioned before, corresponds to a very broadband optical spectrum. So we're talking here about a spectrum which covers from, let's say, more or less 990 nanometers up to 1140 nanometers. The output power for this laser is in the range of 3 watts, giving you a peak power of 2.5 megawatts, which is quite significant. As I mentioned, it's an integrated dispersion compensation, so you are able to compensate almost all optics. And yeah, the system is quite compact, has some kind of separate electronics, and it's fully remote controllable and always optimized by itself to the optimum performance. And you can see the very good beam profile. So coming to the question, how we are doing or how we can do 2 and 3 photon imaging. So as you have seen, the very short pulse corresponds to a very broadband spectrum. And you can see in red that if you take the near-infrared spectrum and do a 3 photon process or a third harmonic, you end up in the range of 300 to, let's say, 398 nanometers. In black, it's shown it's a typical transmission curve of a microscope objective, and you see that there is some kind of transmission, let's say, in the blue part. So you see that after the third harmonic, you are able to detect some light, I would say. If you just compare it to a 150 femtosecond laser, which has a much smaller spectrum, you can see that doing a third harmonic, you end up more or less in the UV, or at least the transmission, the light is not transmitted to the microscope anymore. And this is the main advantage. If you're going with the spectrum above 1080 nanometers, you are able to do third harmonic imaging. And I just want to say, doing this with longer pulses at shorter wavelengths, third harmonic is at least, let's say, it's at least very hard to do. And I just want to show you some examples of what you can do with second and third harmonic imaging. And one example is the high harmonic generation imaging microscopy. And let's say you all know that if you want to do biopsies, at the moment it just takes time. So you want, let's say, in the end, you want to investigate the tissue. And for now, there are options that say you take out of the biopsy, you do some kind of fixation, embedding, slicing, and H&E staining, which typically gives you some time frame in the range of 24 hours or more. If you just use the same tissue and put it under the microscope and do second and third harmonic imaging, as I mentioned, you can see all the cell structures, collagen and elastin fibers, and you can get a picture within less than a second and a complete biopsy overview in 7 minutes. So just this explains how second and third harmonic imaging can be used, especially in terms of cancer detection. And it's a very nice and very fast method to do this. As I mentioned before, it's also possible to do long-term imaging with really short pulse durations, simply because you can use very low average powers here. And I just want to show you some examples which were done with the University of Amsterdam, where they did some kind of analysis of brain tissue, where they could follow up neurons for several hours without destroying the samples. And this is one of the advantages of these very short pulse durations. And with this, I want to come to the famous epic questions. What can we do for you? What can you do for us? So at the moment, we would be able to provide demo systems for testing, and we are able to offer solutions for multiphoton applications, not only in bioimaging. I want to mention that the focus is really on making use of very short pulse durations and what you can do with this and what you can do for us. In general, we are interested in contacts and ideas, and we are looking for collaboration partners who want to, let's say, explore the technology and to move forward with us. And therefore, thanks for your attention.